Tēnā koutou katoa. Welcome to the Geospatial Research Institute Toi Hangarau seminar, the last seminar for this year, 2022. And it's really great to see so many people connect online and have people in the room as well. It's a very interesting and important topic going to be talked about. And I'm very pleased to welcome three speakers, one of whom is online, Cecilia from Manaaki-Whenua Landcare Research, and two of whom are in the room here. We have Audrey Lustig, also from Manaaki-Whenua Landcare Research, formerly from Geospatial Research Institute. Great to see you again, Audrey. And we also have Sharmila. Sorry, I can never say. Thank you, Sharmila. I can never say your surname. We have Sharmila in the room as well. She is a developer at the Geospatial Research Institute, and the topic will be Mātauranga Māori Framework for Surveillance for Plant Pathogens, and it's a project that's been running as a collaboration between the GRI and Manaaki-Whenua Landcare Research, with GRI's been contracted to do some of the software development work that Sharmila does, and so you'll see some of that work towards the middle part of the talk. I'll you don't want to hear from me, so I'll stop talking and hand over to Cecilia, who will start off first. I see you online there, Maria, and I'll hand over to you and then adjust the audio as needed.
Kia ora, my name is Cecilia Latham, and today I'm here to talk to you about our work around the Mātauranga Māori Framework for Surveillance for Plant Pathogens. I want to acknowledge my co-authors, or the other people that are part of this work. So Audrey, Lustig, and Sharmila, who are on site. And unfortunately, I was going to be on site, but circumstances prevented me from doing that. But I also want to mention that another member of the team, Dean Anderson, is also join us online here, and there's also Waitangi Wood, the last member, and she couldn't join, unfortunately. She's tied up with other things, but nevertheless, a big part of the project. So I just...
You've been muted.
Sorry, Sharmila is going to be moving the moving through the presentation for me. So Sharmila, the next slide, please.
Yep.
So first of all, I just wanted to sort of put our research, or this particular research, into context. So the research falls within the National Science Challenges. And I don't know if you've heard of this, but these are cross-disciplinary mission-led programs designed to tackle New Zealand's biggest science-based challenges. And these were set up in 2014 for going over 10 years. And there's 11 challenges, and some of them tackle issues related to health, to the sea, to Antarctic, but our research program, or our research, falls within the... Sharmila, next. This is going to take some time for me to get accustomed to this process, this way of doing things. Our research falls within the New Zealand Biological Heritage Challenge. Next. And this challenge aims to protect and manage New Zealand's biodiversity, improve our biosecurity system, and enhance our resilience to harmful organisms. Next. So the Bioheritage Challenge aims, the goals are to empower New Zealanders to protect our precious environment, to protect by developing a world-class biosecurity system that can detect threats early and act accordingly, and to restore the ecosystems to create a resilient environment to new threats. So the overall mission or the ultimate goal is to reverse the decline of New Zealand's biological heritage through partnerships to deliver step change in research, innovation, technologies, and community and sector actions. Next, the role of the challenges is to connect, rather than delivering the research itself, is to connect a range of partners and existing investments to accelerate progress. Next, and this is done through collaborations, what they call collaborations, and there is collaborations of multiple agents, research providers, and community groups, and so on. And this occur in different areas, but specifically our research that we're presenting today falls within the kauri, dieback, and middle rest theme. Next, Sharmila, please. Yeah, so we fall under the kauri, dieback, and middle rest. So next. The kauri, dieback, and middle rust are diseases that affect our iconic trees, so kauri and myrtaceous species, so things like the buhutakawa and rata. And as these are quite important species, or taonga species in New Zealand, there's been a specific program tackling these diseases has been set up, and this is called the Manaaki-Whenua program. And the aim is to, again, accelerate research related to this diseases by developing new knowledge and tools needed to empower New Zealanders to protect and restore the forest for future generations. The research within Manaaki-Whenua is occurs on the 7 themes, basically occurs on the 7 themes, which with the first 2 of them concentrating in empowering communities. The number 3, 4, and 5, looking at protecting, so research that is related to the disease itself and how to find the disease and how to control or protect the trees against the disease and cure. And the final one, the final 2 research themes are focused on learning more about the pathogen and restoration of affected areas. So next, Sharmila. The research I'm talking today falls under theme 4, which is the integrated surveillance theme within the Manaaki-Whenua program. Next. So the integrated surveillance theme or research stream aims to develop a hapū-centric surveillance framework for plant pathogens by elevating Manaaki-Whenua and the state of the forest into the biosecurity system. And we have 3 main research areas where to address this aim. So the first research area is the development of the Mātauranga Māori Framework for Surveillance, what I'm going to call the MMFS, or you're going to see called as the MMFS. The second research area is the Integrated Intelligence Platform, or the IIP, which Sharmila is being working on, and she'll talk more about in a second. And the last component or research area is the proof of absence model for plant pathogens. And Audrey Lustig will describe a bit of the work that we've been doing within that research area. Next. And so to talk a bit more about the Mātauranga Māori Framework for Surveillance. Next, Sharmila. It aims to, again, elevate Manaaki-Whenua and the state and health of the whenua into the systems, not only the surveillance system, but the biosecurity and also research. And it recognizes that both Western and Indigenous knowledge need to be considered to find to develop tools and approaches and find solutions to address the plant pathogens that affect our native biodiversity. Now, in this case, we are concentrating on middle rust and kauri dieback as our case studies, but it can be extended to any other pathogen or also pests that affect our taonga and the nahere. The approach is based on a co-creation or co-design process because it aims to identify needs and priority of a diverse group of people. And it has recognized that it's critical to develop and maintain trustworthy collaborations between researchers and hapū and iwi and other local institutions and government agencies. To put this framework into practice, we are developing the 2 online tools that I just mentioned before, and we'll go into more detail in a second. And key to the framework is the distinction between data custody and data sovereignty. And I'll go more into that in a second. Next. So the framework classifies any science and research activity or management plan for plant pathogens along these four different quadrants here. So on the left-hand side, it's more an approach where a single knowledge system is used to find solutions to a complex problem, such as the plant pathogens that I talked about. On the top, we have an approach where solutions are found using only Mātauranga Māori, or on the bottom side of the quadrant is where the Western knowledge is the leading authority to find those solutions. And then Mātauranga Māori has oversight of the research and might consider impacts on the Māori worldview. But Western science is the approach used. And in this case, the sovereign authority of data and information is recognized. On the right-hand side of this graph, we have an approach that combines both knowledge systems, either on the top being Mātauranga Māori-led, but also with support from Western knowledge, or the opposite in the bottom part. In both of this right-hand side of the graph, the idea is that both knowledge systems act as valid ways of obtaining information or doing research, and they sort of complement each other. And in both cases, there is cultural license, and the research is also socialized with hapū and Manaaki-Whenua. Next, and within the Mātauranga, we recognize that any of those areas are valid ways of doing research, but we want to work towards the right-hand side of that graph. So I talked before about the Mātauranga Māori Framework, making a distinct, important distinction between data custody and data sovereignty. And we do this through what we call the biodiversity management areas. So Maori data sovereignty refers to the rights and interests that Maori have in the relation to the collection, ownership, and application of Maori data. And Maori data being any information or knowledge, either in a digital or that can be digitized, and that contains any Maori content or is associated to Maori. And this includes data and information about their lands, water, and taonga, regardless of who has collected it. Now, on the other side, or on the opposite side, is the data custody, who refers to the curators, users, or those who have acquired the data. Now, within the Mātauranga Māori Framework, we give effect to data sovereignty by recognizing that any data is place-based and anchored to special units called the biodiversity management areas. And these special units represent tribal committee areas, and they were gazetted during the 1940s and 50s under the Maori Social and Economic Advancement Act. So the key things about this areas are they are recognizable land units, that they are recognized under an official act, and the description of the boundaries of these areas have been documented in the New Zealand Gazette, which is the newspaper of the New Zealand Parliament. These are traditional areas where Tangata Māori manage the biodiversity and activities, and they are contiguous across New Zealand. So there's about 400 of them across New Zealand, and there is no gaps. Basically, they cover the whole of the country. So this is a way that we've identified that can be used to recognize the sovereignty of data by anchoring, or the provenance of data by anchoring that any data or information or taonga that is collected to the landscape or naturescape where it came from, being the BMA. Next one. And that brings me into our second research area, which is the research area 2, which is the development of the Integrated Intelligence Platform, which is an approach to support Indigenous data sovereignty priorities. Next, please. And this is just sort of a schematic of how the IIP works. Sorry, the IIP being the Integrated Intelligence Platform. So we start with a tree. Sorry, it's hard to show, but if you can see on the right-hand side, there is a tree and data collected at a place from a specific site or tree. And that data generally collected by agencies, being an example of that, MPI or any researcher with interest on addressing a research question. Generally, the data that gets collected gets taken back to the office and by this custodian, and it's stored in the servers or the personal computer of this custodian that collected the data. Now, the data might be shared internally with other members of the organization or externally with other researchers. Now, within the Mātauranga Māori Framework for Surveillance and the development of this integrated intelligence platform, what we aim for is to bring that data back that has been stored into within the custodian servers or storage places back to the land, so back to the place where it was collected. And we do this through assigning this, what we call the BMA reference, or linking the data, each piece of data with that polygon for that BMA. And by doing that, we give access to that information to the sovereign authority, which is the biodiversity management area. Next one. So the Integrated Intelligence Platform is a spatial data infrastructure that allows users to store and share data about Aotearoa's biodiversity and its threats. So it's not just plant pathogens, but anything, any data can be stored on it. And it's a novel system that recognizes and gives effect to Māori data sovereignty by connecting data and information to the place where it originated. So in this case, the BMA. It can empower Indigenous people through timely access to existing and new surveillance data information. As I said, once the data gets uploaded to the IIP, it gets linked back to the BMA, and that allows access to the information in a timely fashion. And it provides a mechanism for custodians to demonstrate the least requirement of cultural license by recognizing the provenance of the data and information. Next, please. A key entity within this whole framework, and particularly within the IIP, is the Tangata Kokiri. The Tangata Kokiri acts as the linkage or the person that facilitates the communication and the trust between the custodian and the BMA or the sovereign authority. So it's a person that is identified for each BMA, and within the platform or the IIP, the tangata kokiri has access in perpetuity to any data associated with the BMA. The tangata kokiri, this role, can manage access, permissions, and controls for individual Mana-Whenua, so individual people, and this is done within the parameters that have been set by the custodian. So this recognizes data integrity and privacy. The tangata kokiri can also facilitate the establishment of cultural authority agreements. So we see it as a way to encouraging conversations between the tangata kokiri and any researcher or agency that wants to do research within a given BMA, and they can talk about this or agree to this cultural authority agreements in which the parameters can be set as to how the research can be done within that ROHE or BMA. So in this case, they ensure that the hapū or iwi values and the Mātauranga Māori specific to the ROHE are considered when designing and implementing surveillance in the targeted areas. And I think that's all I had to say. Sharmila, the next one.
Yep. Thank you, Cecilia.
So now Sharmila is going to talk a bit more into the nitty-gritty of the IIP, but I just gave an overview.
Yep. Thank you, Cecilia. So yeah, so IIP is our data storage platform, which is custom built for this particular project needs. So this in this image shows the BMAs of Northland. All over New Zealand, there are more than 400 BMAs. And we wanted to design a data storage platform which anchors data to its place. The places are called BMAs. So the relationship here is the data is anchored to a BMA. BMA has Mana-Whenua. Mana-Whenua have their tangata kokiri, who represents that particular BMA. So whenever a data is linked to a particular BMA, the kokiri person gets full access to that data and he can manage that data. And the Mana-Whenua can get access to this data through their tangata kokiri of that BMA region. So we wanted to develop a spatial data infrastructure which would do all these requirements. So, and I will show you a demo in a minute. So these are the permissions that is set for the resources. So this is simply defining who can do what. For example, who can view the data, who can download the data, who can change metadata of that information, and also who can edit the data for this particular layer, and who can edit styles, and also some more advanced privileges like who can update, delete, and change permissions. So say, for example, when a particular data is shared with linked to a BMA, the kokiri person gets access. So in this example, you can see, Sharmila is the data custodian, and this particular kokiri is the one where this data was linked to the BMA. So to develop this spatial data infrastructure, I used an open source web framework called GeoNode. GeoNode is useful for storing and sharing geospatial data. It's a geospatial content management system, which is also extendable. So you could customize it for your project needs and also to suit your organization. So that's what I used to develop this data storage platform. So far, the implemented functionalities are the user registration process, which is customized to include a set of questions aimed at describing the variety of platform users. and also addition of some custom metadata information, which is very particular about this project, and also sending out automated emails through this platform, and storing and sharing of data among different users and groups. And also we implemented some roles and permissions just to ensure so that we have better control over the data access, and also to ensure the data security and mainly Maori data sovereignty. For future work, we would like to add the component of location masking with data encryption. And this feature, it has been developed by our researchers from University of Auckland, and that will be added into our platform as an add-on. That's our plan. And we are also working towards the deployment of this platform, and it will be tested by our end users and collaborators before public release. I will show, I made a demo video. I'll just show you in a minute.
Hello, everyone. This is our IAP GeoPortal. I would like to show you how to store the data and share it and link it to a BMA and how to set permissions to it. Let's sign in.
Sorry.
Hello, everyone. This is our IAP GeoPortal. I would like to show you how to store the data and share it and link it to a BMA and how to set permissions to it. Let's sign in. Now we are signed in. We will upload some data. We will upload surveillance data. Let's wait. Okay, it's uploaded 100% now. So it's here. Yeah. We will add some metadata information to it. For abstract, we'll just say KD surveillance and attribution. We'll say this. And maintenance frequency as necessary. Fetex keywords, let's say plant pathogen. Restrictions, formal permission to do something. And we will link it to a BMA area. We know this is from this region, which is BMA 5. So we select BMA 5 here. And this project is funded by Audrey. Let's save this. And this is the disclaimer showing, okay, if you choose one BMA, only one, that BMA, the Kokiri will get access. If you choose more than one BMA, then different cookeries will get access. Then it will be seen by the people of that region. So it's saved now. Now let us set some permissions, layer permissions. Okay, so by default, anyone can see. We don't want that. We want only the permitted people to see it. So by default, the owner of this data, which is Sharmi, can see it. And this cookery 2 is the Tangata cookery person of BMA5. So by default, he gets access to it as well, to view it and to download it. And we don't want anyone else. So let's take this out. And yeah, so the data custodian can change metadata for it, and we don't want anyone to do that. So we won't include any people here. So the same with editing the data, the same with styling the data, and who can manage it. So it's both the data custodian and the cookery of that particular BMA. Let's apply these changes. It's saved now. So thank you.
So that's a 5-minute demo of the tool and how it operates. I will give it over to Audrey. Thank you.
Thank you. So I'm going to move on to a third part of this talk, which is about how we're going to apply all this in practice and what it would mean for Manaaki-Whenua or community surveillance and how they can interact with all the methods that we presented. So I've decided today to take as an example kauri dieback. So I wasn't sure what was your background about the disease. I have this introduction slides here. So kauri dieback is caused by a soil-borne pathogen that will cause root and color rot that can create bleeding around the trunk of the trees that will lead to canopy thinning and eventually to tree mortality. Yeah. Oh. Can you hear me again?
Can you hear us more clearly now?
There is a radio.
Yeah. Feel more comfortable using this?
Oh, so I'm more. It can. Oh yeah, better. Thanks. So the disease was first reported in 2006 in New Zealand, but we imagine that it's been here for way longer, and it's been since then confirmed in almost most of the natural range of kauri and kauri dieback. So the pathogen can live dormant in the soil for quite a while, up to 15 years. So we'll be at a location without generating any symptoms in the tree, which make the detection of the pathogen extremely difficult. We don't know much about how it spreads. Looking at other phytophthora, we believe that it's focal in nature in the sense that it will spread root to root or through subwater transport. There is long distance dispersal happening, and this is usually through human activity when we move soil from one place to another, either by just walking a track or going trapping pests in the forest or through animal vectoring. We currently don't have any cure for the disease, so management practices include closing healthy forests, quarantining area that have been infected, or if you go in the North Island, you will be asked to clean your shoes before you enter a forest and clean it when you come out of the forest. So initiating all those hygiene measures. We currently don't know where the disease is and where it isn't. Partly because it's extremely difficult to initiate forest-wide surveillance, and there has only been really two forests that have been extensively surveyed, and we still don't even know within those forests where the pathogen is really. But also because there has been a lot of disconnect and little collaboration between the agencies initiating surveillance, and because we have a lot of lack of resources, it takes a lot of people to go and initiate the surveillance and lots of funding. Often those funding or even any program that I have initiated surveillance have left apart any Manaaki-Whenua from their program. So we hope that by implementing the Mātauranga Māori Framework, we can help initiating those collaboration and include Mātauranga Māori and Manaaki-Whenua really from the start of any surveillance program. So ideally what we want to do is when you go to a forest, you will be able to map within the forest where your pathogen has been detected and to, well, where the disease has been detected. And for detecting the disease, you need to find both the pathogen and to observe the symptoms. That's, you know, that here on that bottom right corner here, and you will start mapping the severity of your disease. It will occur that sometimes you will only see symptoms and not detect the pathogen, so you're quite unsure what's going on here, and that could be false or true positive, and the symptoms are not specific to kauri dieback. They could be caused by many other factors. Water shortage is a big one, so it could be that it's simply other factors affecting the trees. You could have detection of the pathogen and observe no symptoms, so it could be that the tree hasn't developed the symptoms yet, or simply that you found an asymptomatic tree, meaning that there is some resistance happening in that particular area, and that can become a very interesting place where you could start looking at solutions. But where we're really going to focus today is on those areas where you have no detection of this pathogen, no symptoms. So you're looking at a pretty healthy forest. And you believe that the pathogen is absent. But if you don't detect the pathogen, it doesn't mean that it's actually not there. It's just that there is some probability that it's not there. And maybe it's just that it's there and you haven't looked enough, that you haven't detected it, or that it's actually absent. And one of our collaborators here, Dean Anderson, has been developing these modeling tools that help quantify this probability of absence. And it will include information about the amount of surveillance you do on the ground and your distance from known infestation. I'm going to get into the details of this shortly. The data we work with, it's been mainly relying on field surveys. So it's people going in the forest, collecting soil sampling, looking at the forest and visually assessing those trees. We also do work with manual interpretation of aerial imagery. So it's actually a person manually processing each individual tree that is that looking at this picture and developing what we call a ground health assessment. So you score this tree, I think from 1 to 5, 1 being a very healthy tree, 5 being a dead tree, and anything in between. So it's a method that comes with its limitation in the sense that it's really relying on the interpretation of the person manually processing those imagery. Unfortunately, at the moment, this is the only data we have that can be available across the full North Island. We know that there are more accurate methods that are coming up, but those data haven't been shared with us so far. And so we are not actually sure that we'll be able to use it in the near future. Near future being the groundwork starting now for this season. The considerable cost of going on the ground, and as well the risk of spreading the disease when you go on the ground and do all the soil sampling needs that you really need to target the area where you go and prioritize your site. That means that you would like to know which part of the landscape are more conducive of the pathogen, or where you would like to invest more your surveillance. So that's the work that we've been initiated with plant and food research and pathology experts over there. We've been trying to map the distribution of the pathogen. At the moment, it's rely only on experts' knowledge, and we hope that during the year we'll get more quantitative relationship. But what Nari Williams, that you see on the picture here, has been doing, she's been associating a risk of pathogen occurrence to the slope, the amount of water you find on the land, the distance from road and tracks, the distance from trees showing canopy decline. We use this aerial imagery as an indicator of where the pathogen is present on the landscape. This is not verified. This is a really strong assumption in this route map, but again, it's not because trees show sign of decline that it means that it's caused by the pathogen, but this is the only information we have so far. And we hope that by the surveillance we're doing this year, we'll be able to find more correlation between all those variables and the risk of the pathogen actually occurring on the land. So if you merge all this information, what we get is this, what we call this relative risk map that indicate on your landscape, in your forest, where the pathogen is more likely to be present. I apologize for those map people. There is no indication. I've been trying to hide where we are here. So that's why you don't have much information on those maps. But the idea is really here. If you can show those maps to Manaaki-Whenua as an indication of how healthy the forest is and where we could start initiating surveillance on the ground. So at the moment, because we work on such a limited resources, financial and human resources, it's impossible to implement forest-wide surveillance. So this year, what we've decided is to actually implement two strategies. The first one that you see on the left is to target very relatively small zones on that landscape, centered around those trees that start to show a sign of decline, and to try to confirm whether the pathogen is there or not, and if those first sign of disease are really caused by the pathogen or by other factors. The other strategy on the right-hand side is that we know we have an historical detection of the pathogen and a tree showing symptoms, and we try to delimit the disease fronts. And so we use that model that is called proof of absence modeling that will help us declare with some confidence that an area is free of the pathogen or not, and will help us refine the surveillance we'll do next year. So how does this model work? I would like you to focus on the middle column here. So we're using surveillance data. So the amount of surveillance you do on the floor, on the land. When I say the amount, is where you go, how often you go there, how many samples, how many trees do you sample in the landscape. We use the relative risk map that I just presented because the probability of finding that pathogen in the landscape is not equitably it's not the same everywhere, but it's easier to say it this way. We use what we call a probability of detection. So we know that the soil sampling are imperfect at the moment. We have some difficulties in understanding how reliable they are. So this is another part of research happening at the moment, trying to understand how reliable are those soil sampling. But most importantly, this modeling rely on Bayesian modeling and use what we call prior information. So this is the prior knowledge we have about the landscape being not infected by the pathogen. So as a quantitative ecologist, I will just go and we look at how many trees are starting to show sign of decline versus how many are healthy, and we can come up with some numbers and include that in the model. But collaborating with Manaaki-Whenua, we can actually start including other information about that relates to their own knowledge about the landscape. And they know their forest really well. They know what's happening. They use Mātauranga to design, to quantify how healthy is the forest. Quantify is maybe not the correct word here, but they can tell us whether an area is more healthy than another one. And that prior information allow us to elevate the Mātauranga Māori within the modeling framework. It's a really important parameter from that model, and we will really rely on that practitioner knowledge and include that in the model so that it will influence that probability of absence. This is really work in progress. Again, this is one of the first case study that we've been running with our first Manaaki-Whenua group. And what we're trying to establish with them is how many tree they need to sample in an area and how many samples they need to take for each tree to be then confident and say, this area where I focus my surveillance is free from the pathogen and I'm confident about it. And early results suggest that it's going to take a lot of surveillance effort. So in short, what we're doing at the moment is engaging with those Manaaki-Whenua. We have 3 area that we target, and those biodiversity management area that Cecilia described at the beginning of the project. We collaborate with Andrew McDonald from Geospatial company who's generating the aerial crown assessment for those different areas. And then we'll link them with those biophysical data to generate the relevant GRISS map that I just showed you. We then meet with the Manaaki-Whenua, present them those map and establish together where they would like to go in the landscape, where are the area where we can't go. Cecilia has been working with Arienti to train some of these Manaaki-Whenua to actually do the field survey and how you will do it. What do you need to look at in the landscape? Where do you need to go? How to collect the data? We discussed the model assumption and asked Manaaki-Whenua to then help us recalibrate the model with us and provide them the sampling location for Manaaki-Whenua to progress the surveillance across their forest. Ideally, we're not quite there yet, but I hope we're going to get there at the end of the project, is that all this data will be then stored within the platform that Sharmila is developing, and that will be able to extend the work that we do with 3 Manaaki-Whenua group at the moment to 8. And the idea is really that the surveillance data can be easily accessed, easily shared, while respecting and giving effect to Māori data sovereignty. Obviously, the surveillance that will happen this year will help us refine our model assumption and refine our relative risk map so that we can be more confident about all the tools we are developing. So it's really a co-development process at the moment, and we are in the process of getting additional funding so that instead of having to limit us to only look at certain part of the forest, that we can initiate forest-wide surveillance program. But that's really thinking in the very long term, where we would help initiate the surveillance program, and then everything goes in the hand of our Manaaki-Whenua, and they will implement the surveillance program over the year. And I'm going to stop here. That's the end of our presentation, and I hope you enjoy it. Thank you.
Thank you, Audrey, just now, and Sharmila and Maria earlier on. Really fascinating and comprehensive talk about all the great work that's happening in this important area. We have about just under 10 minutes for some questions. I'm sure there are some. So I'll invite anyone in the room to ask. I'll bring a microphone to you. Anyone online could either post it in the chat or feel free to speak up. So let's see if this works. So any questions? Luke.
Hi, so with the tangata corporate, if they want to share the data back to people interested in the BMA, such as the hapū or something similar, how would they provide the data?
So the understanding is that if they belong to Manaaki-Whenua, then Manaaki-Whenua can get their access through tangata kokiri. Okay, but if it is someone outsider, then we believe there are some process that would happen out of the platform. Okay, so that the kokiri person can contact the custodian. So these kind of yeah. So this is not happening through the platform. These kind of things has to happen out of the platform. So if they belong to Manaaki-Whenua, it's direct, okay, because they have the sovereign authority. Okay, yeah.
Thanks, and
My question is for Audrey. That's a really interesting model. I was wondering that you added some anthropogenic agents to check how the spread of that disease is moving around the forest. Are there any elements with the fauna, like bird migration or anything else that are affecting the disease as well?
Currently, don't know. So we focused on the main parameter that we know will affect the spread because it affects other phytophthora. I wouldn't think that birds migration will affect this pathogen in particular, but obviously we're certainly missing things. One thing that comes in top of my mind is to which extent other species in the forest will affect the root-to-root spread. And this
is it like something like possum or other small?
No, it's actually the pathogen moving from
Inside
one
the-
root
okay.
to another in the soil. So we don't know if other species can, act as a gel. So you're from one tree, you have a different tree, will affect this tree and then go to another
It's
carry.
more like a chain reaction, is it?
Yeah.
Okay, thank you.
we have a question online from Chris saying, thanks so much. I'm really interested in the intersection of Mātauranga Māori and Western science. What tensions came up that perhaps were difficult to solve? Maybe that's another question for Maria, perhaps.
At the moment, we're working through this. So we've been working very closely to the other themes within the Naraki Takitaki, and there's really not, the Western scientists are really keen and open to it, and Maori. So the way we work through this is through the this cultural agreements, where a scientist introduces the research to the Maori authority, so the kaitiaki in each that has been, or the from the BMA, and they go. There's quite a there's a conversation as to how the research can occur and how much Maori involvement there might be. So at this stage, and because it's all been done through under the NRT umbrella, it's been quite a good process, and it's yeah, once we start to try to extend it beyond the NRT umbrella, it might prove more challenging, as you say. But basically, I think, minimally, as we said, we are trying to encourage any research that is being done to at least recognize that data sovereignty and at least give that option back to the people, the Manaaki-Whenua, to access that information and be able to use it to manage their lands or make better decisions. And we think that's, the minimal requirement that should be completed or done for any. That's the basis of the Mātauranga Māori Framework, and from there build on to any type of collaboration. But I think also very importantly is the fact that we are identifying this tangata kokiris. It will make those conversations easier to occur, because I know, at least from within Manaaki-Whenua, in many instances, it's quite difficult. Researchers don't know who to talk to, and I think that's at least a place to start as to how those research programs can move forward in specific areas. I don't know if I answered that question.
There's another couple of comments just coming in online. Again from Chris, thanks so much, appreciate it. I'm thinking operationalizing Mātauranga in research and planning ahead, data sovereignty discussion, quite a great insights for me. And Tangata Kokiri seemed like a good way to have a hub person. Thanks for the comment, Chris.
With regards to the operationalization, sorry, as I think a very important part of our Mātauranga Māori is this this agreement, and there is this cultural research agreements that we've put in place to basically to move research forward that can include both science, I guess, or those knowledge systems. Anyways.
Thanks, Maria. And from Rachel, thanks for a brilliant talk. There seems to be a great potential for many kinds of environmental data to be integrated within this framework in future. Would the framework also be compatible with data on wide dispersing or migratory species that might be connected to multiple BMAs? Sharmila has an answer here.
So even now, it can be data can be connected to multiple BMAs. Just that the custodian has to be, aware that then because if it is sensitive data, or if they want to share it with only one BMA, then they should post, upload that data about that particular BMA. If they don't mind, other people from different BMAs seeing it, then it's fine. Already the system supports multiple BMAs as well. So if you have data from different BMAs, you can still upload, still link to different BMAs, and it will be seen by different tangata kokeris and the corresponding people. Yeah, it's possible.
Thank you. There's 3 questions. Let's start over here. Maria first.
Thank you. I have a question regarding data collection. Since you are integrating these 2 different sets of data collection, are you going to have like a standard way of collecting the symptoms or the findings for the trees from both sides, the Western view and the Maori, or are you going to have just a standard way of adding this information into final diagnosis or the way that you're going to analyze models, etc.?
Cecilia,
Sorry. Yep. So, yep. Yeah. So at the moment we're working with about 10 to 14 BMAs, and we've developed some survey forms, and the survey forms have and these are survey forms that are gonna, standard forms that are being used in the field to collect all this data. At the moment, we're kind of concentrating on kauri dieback, but the middle rust is coming, form is coming, and those forms, some of the information has been it's required by researchers in the other themes. Like, as I said, there's a number of themes, and sorry, might put my video. There's a number of themes, as I said, in Naraki Taki, there's 7 themes, so all the data is collated back to them, but there's also data that is purely Maori, of Maori interest. So each one of the Mana-Whenua is, at the moment, is using a standard form, but that form can be extended to include things that they want to collect for themselves. And as I said, that data just goes back to be stored in that platform or will be stored in that platform. And that's data that is just belongs to them, and it just they can share if they decide to do so. But yes, there is some, that's more the Mātauranga data, and there is also the Western scientists data that is being collected for the researchers. And as I said, there's been an agreement that that's okay, between the researchers and the Manaaki-Whenua groups.
Running low on time, but there's two quick questions. Hopefully quick.
I had a question just about your model development. So I was sort of looking at you've got your area surveys, and I know you have got all the data that you want from this year, and you've got it down to a resolution of about 10 meters. And I see your model looks like it's got a 10 meter thing as well, but it's not quite yet taking the landform into account, or maybe the waterways are, from what you're saying, higher risk because of the transport of the disease through those cities. Is that where you take? Oh, well, I guess my question's around is where are you taking it? Where do you see the best advances are for your next steps?
I guess it's twofold. You're right. We're having a challenge with the spatial resolution. So all those tools have been developed for actually pest management and have been adapted to the plant pathogen, and downscaling to the level of a tree has been a really big challenge to us. We've also been limited by the biophysical data that we can access at the moment, and we've limited ourselves to anything that is available online and freely available. So it's this digital elevation model, and most of our slope model and subwater model rely on this map, which is provided at the 25-meter resolution freely. So we know we could use more grand scale data set, and they exist. Can we access the data for every BMA at a national level is a bit question. So for now, we've been trying to be consistent across our BMA, and it's been proven to be enough so far, but it's very early stage. And if that's an issue, we'll need to get access to those more fine scale data sets. Yeah.
We're out of time, but we're going to allow one very last question.
Oh yeah, kia ora tātou katoa. Yeah, mine just follows on from Ria's really in terms of data collection. So one is who collects the data? Is it kaitiaki that collect data? And the other one is about, Cecilia talked about forms. Is it like a paper-based form and then you put it into the computer later, or do you actually have field tools, digital field tools for the collection of that data?
Did you hear, Cecilia?
Yes, the second part, not the first part. Sorry.
Is it the kaitiaki collecting the data? Who's collecting the data?
So the data is being collected by the Manaaki-Whenua. So yes, there is basically within each BMA there is groups of people that are more in. There is organized. So the hapū is the one we're working with, hapū groups, and they have certainly like environmental teams working, and we are. So it's and this goes back. It goes a bit beyond our integrated surveillance. So it as I said, it involves all the other teams within Naraki Taki, and so we've set up this sort of agreements, and it's this environmental groups within each hapū that are doing the work, the data collection. And yes, we are using digital tools. So basically, we provided, we're using the Survey 123, which is one of the ArcGIS apps, and we can basically, we've developed the survey forms using the survey 123. The data goes into ArcGIS Online, and then it's going to get transferred into the IIP once it's running. Ideally, we would like to not have to use the ArcGIS, but build the app the survey app, so the field forms directly into the IIP, but that's another iteration. It's not going to happen right now. I don't think. We first need to put it online. But yes, it is directly stored online. It's a digital form, and yeah,
Oh,
I don't know
okay.
if that's
No, that's great. Thank you, Maria. And we're 2:05, so we're 5 minutes late. And apologies for that. I know that other people have got things to do, and I think it's just a reflection of how interesting this topic is, and we could just keep asking questions all afternoon really, and exploring all the issues. I have a few questions too, but I won't ask them. Yeah, no, thank you so much everyone for joining us today for this last seminar of the year. What a great way to end the seminar series for this year. Thanks everyone for joining online. We have got a recording of this, so hopefully we'll be up on our website at some point. And we'll wish you all the best for the end of this year, and see you in the series starting next year. Not sure when, but starting next year in February. Thank you, Maria. And so yeah, thank you once again, and let's thank the three speakers who are all excellent for their talk today. Thank you. So see you again, and thanks for joining online, Maria.
no problem. Thank you.